Hi, everybody. Welcome. My name is Chris Kinzel. I'm the program coordinator for the Westchester Biotech Project. And if you're here for sustainability, you're in the right place. And if your lab doesn't look like this yet, it's not too late. So here at the Westchester Biotech Project, whether you're new to our group or you've been with us for a little bit, we're conveners and bring together thought leaders, experts, resources in the region, connect these resources in the hopes of creating opportunities for collaborations and advancement in our ecosystem. So the biggest thing to take away is that we are an organization that operates based on our community and their needs and their participation. So without our participants, such as you, we don't really get much done. So we have our LinkedIn, YouTube, Facebook, Twitter. Welcome you to join us, follow us, get involved. We would love to talk. Just reach out. And Joanne Gear, our executive director, is here with us. Joanne, you there?
Well, good morning and welcome everyone. So in addition to bringing people together, which is pretty exciting, we started in the spring of 2017 and have done quite a bit of convening, which has been fantastic. But what's starting to happen is that real action is growing out. So for example, we are on November 1st, we'll have a meeting with our roundtable for educators and employers. And actually, two of the people on our call today are participating with multiple different colleges, universities, and companies to develop a curriculum for basic lab skills, like ranging from safety to engaging with sustainability to lab notebook practice, aseptic technique, and so on. So it's not just that we're getting together, which is good enough, but that we're actually seeing a lot happening. So I'll pass it back to you, Chris. Thank you.
Cool, thanks, Joanne. And on that same note, so this sustainability focus is a part of that certificate program. And so keep an eye on the progress with that. And this list right here, as I mentioned before, our community partners and alliance partners, we form relationships with a lot of these other organizations in the region. And, you know, it's hard to get big stuff done alone these days. And so without our partners, we kind of just be treading water. So big shout out to all these guys and our events, our actions. So we have a innovation and research is kind of our yearly gathering for the region. We have another one coming up in the spring. I want you to be aware of, and a tauopathy colloquium that we'll be putting together around the same time, along with other events and projects. Today we're here to focus on sustainability, and so as we move forward, I know that the green talk is a big topic everywhere. Everyone's heard of it and is aware of it. But as a researcher myself in the past, I wasn't really aware of the impact and effects that this could have in the lab and cost in terms of saving money, operating efficiently in the lab. And so today we have our alliance partner, My Green Lab, who is represented by Erika Daley. We've been friends with them for a while, and they're a great organization that's nationally trying to raise this awareness and sustainability focus in research. And also Stephen Harris with Purchase College. So I'm just going to introduce our speakers real quick and then pass it over to Stephen. Erika is a key member of the leadership team at My Green Lab. She manages programs throughout North America, including the Green Lab Certificate Program and the Green Chemistry Curriculum Guide. She has garnered experience in the design, implementation, and management of Green Lab programs in companies and universities throughout the US and Canada to deliver safe, more efficient research and teaching spaces with a lower environmental impact. Stephen, Dr. Harris, uses genetics, genomics, and bioinformatics to study the evolution, ecology, and behavior of natural populations in response to environmental change. He has performed fieldwork in urban ecosystems in New York City, coral reefs in the Florida Keys, and Belize, and tropical forests in Indonesia, which has led to his passion for democratizing science by using the latest innovations in biotechnology to build portable, low-cost, and user-friendly sequencing labs for research and education. And it's exciting. Stephen right now is actually just got his lab set up. He has a teaching lab that he's implementing some of these sustainable practices into, and it's really the end game of what BioGreen Lab's efforts are. So, Stephen, if you're ready, I'll pass it over to you.
Hello, everybody. I'm just pulling up my slides here. So you should be able to see my presentation full screen. Thanks for the introduction. I'm really happy to be here. As Chris mentioned, I'm an assistant professor at Purchase College, and just setting up my lab. So you can probably guess I'm new. I'm just finishing up my second semester as a professor. And it's really interesting at this time to be setting up my lab to start thinking about ideas about lab sustainability, because I'm in the process of ordering equipment and getting things ready currently, and all of this is going through my mind every day as I place new orders. So I thought I would give sort of a little narrative or a little story about my research and my background and the type of lab that I run, and then how lab sustainability and methods to work sustainably fit into my everyday work and thinking. So you can see on the picture here, this is a map of Manhattan. I don't if you're not familiar, that's Manhattan. On one side is a representation of what Manhattan looked like before European settlers arrived, and on the other side is a current state of the urban infrastructure. And I sort of wanted to zoom out to a big picture first. And so a little bit of introduction. I think Chris introduced me well. I'm an evolutionary biologist. I use genomics, basically studying biodiversity and how animals are adapting and changing to anthropogenic changes, human-driven changes. Everything from snapping shrimp in the Caribbean to the country mouse versus the city mouse to bats in Indonesia and coral reefs in the Belize. But a lot of my work is interested with the Anthropocene, and this is this new epoch that scientists are increasingly saying that we've entered into, a time when human changes to the environment are leaving permanent records in the geological record. More than 50% of the world's human population will be living in urban areas in the next decade, and that number is just going to increase more and more. And we have this aspect of the Anthropocene called the Great Acceleration, where lots of metrics that measure human impact on the environment are just increasing exponentially. So a lot of this, from my research point of view, is how do we respond to these changes, or how do native flora and fauna respond to these changes, and how do we move forward and create an environment where we can have native ecosystems living alongside the changes that human beings are imposing. So I guess another definition of the Anthropocene, the human epoch: human activity has been the dominant influence on climate and the environment. So this is sort of a blanket definition. Now, for me personally, I try to find questions within this to study, and looking at urbanization in New York City in particular is a great way to do this. A lot of my research, the bulk of my research is focused on urbanization in New York City. So on the left, right, is this representation of Manhattan in 1609 before European settlers, and on the right we see what it looks like today, this mass of human infrastructure, inhospitable urban matrix with pockets of green space dispersed in between there. Now, looking at that, the idea, there are animals and plants that live in this urban space, and the question is, what animals and plants are they, and how are they responding to this massive change that humans have created. And the first idea is separating the types of animals that would be occurring in this space. And in terms of urbanization, we think of three big topics: urban avoiders, so large predators or organisms that just don't do well in urban spaces. So again, like I said, large predators, or this could be smaller organisms that rely on thick forested habitat. Then there are urban exploiters. So a lot of times when I tell people I study mice, they think of the house mouse or the Norway rat, cockroaches, pigeons. But these are organisms that have sort of evolved with humans and follow us from place to place, utilizing the human infrastructure. What's really interesting are these urban adapters, organisms that still live in the green spaces in urban areas, but seem to be doing well, right? So they could live fine in a native forested habitat, but they do just as fine in urban areas. So on the right side, you see a picture of the white-footed mouse on the bottom right, which does well in the Catskills, but also thrives in Central Park in the middle of Manhattan. So the idea is, how are those organisms adapting and changing? And that can give us some insight into, as humans disturb more and more human habitat, what animals are going to do well, and what can we do to help animals succeed as we change the habitat. And the question really is, the urban ecosystem is not, or urbanization is not devoid of ecology. The urban ecosystem itself is a unique ecosystem that has its own selective drivers, and animals and plants can be adapting in unique ways within the urban ecosystem. So on the right side, we see a typical urban setting. The green space, let's say, contains mice in the middle, and around, we have barriers to dispersion like roads and cement. We have air pollution, light pollution, sound pollution, all these things impacting mouse populations living in these green areas, and then potential adaptation to deal with these novel selective pressures. The white-footed mouse, Peromyscus leucopus, like I said, is not the house mouse. He is a native rodent to North America, about 60 million years diverged from Mus musculus, the house mouse, but it's found across most of the eastern North America. A very generalist species, does really well in old growth native forests, but as well as urban settings. And the question is, well, why is that? What is happening to these urban populations of mice that are allowing them to adapt and do well? Right? Is there novel mutations in genes that allow them to deal with selective pressures? So one of the first things that I've done in this research is found that yes, urban mice are different than the rural mice. So what you see here is a map of the New York City area. It's a little hard to read, but you can see Long Island stretching off to the right, and then above the red line, you can see Manhattan down here towards the center, and then this is the Hudson River, not filled in, but the rest of this is New Jersey and New York and some of Connecticut. But this map is showing land use. Red areas are human infrastructure, green areas are forests, brown and yellow areas are agriculture, and each dot represents a mouse population that I've done genomic testing on. And what I'm showing you is the genetics separating out some of the populations. The red line is separating out Long Island mice are different than Manhattan mice and mainland mice. And then on top of that, at a different level, when we look at the genes, the black dots represent urban populations and the blue dots represent rural populations. When we look at genes, the rural mice form a separate population from the urban mice. So that's initial evidence that not only are these populations separated from each other, but that the urban mice are potentially adapting, and positive selection is causing unique genes to evolve, microevolution. And I'm not going to get into a lot of the details, but just to give you an example, again, I'm showing you an urban site compared to a rural site. satellite image plus a land use image. So if you look at Manhattan, Central Park is the only real place in Manhattan here that is not human infrastructure, versus a rural site where it's all green, blue, or green, yellows and browns, right? So I'm comparing these two sites. And what I found were mutations in genes of urban mice that had to do with a particular set of phenotypic traits. Those traits mainly involving metabolic processing of novel dietary resources. So the idea being, maybe urban mouse populations are subsidizing their native diet on seeds and invertebrates and fungi with human food waste, causing positive selection on genes that have to do with cholesterol processing, modulating high fat in your diet, smooth muscle contractions, and insulin secretion. So there might be positive selection on genes dealing with high fat diets from mice eating human food subsidies. So this is just an example of how organisms could be adapting and evolving in response to these human-driven changes. So this is an idea of where my head is at. My research, what I'm thinking about all the time was how biodiversity is responding to these human-driven changes. And at the same time, I've traveled around. I really try to bring modern biological, molecular biology techniques to students and populations that are maybe scientifically underdeveloped. So I developed these traveling lab suitcases that I've taken to Belize, where we've done testing of mislabeling of fish fillets in Belize down here. I've taken kits to Indonesia, where we sampled bat populations. And the idea of trying to train the next generation of scientists. Now, after working with the Westchester Biotech Project, I started really thinking, and then this webinar in particular, started thinking about, I've been so concerned with the impacts of the Anthropocene and training people, I haven't fully considered my scientific impact on the environment. And that's something I study, but then I am wasteful and in a lot of ways. And now that I'm setting up my lab, so this actually picture I took yesterday. So this is the first two students working in my lab space, doing genome sequencing of fecal samples from golden lion tamarins, this endangered monkey from Brazil, and songbirds in the Northeast to look at mercury levels. So you see the lab is a little bare, but as we're building it, I'm starting to think of ways that I can do it in a sustainable way. So some of the ideas and options that I'm thinking about over the next few weeks and months as I set up my lab, one with electricity. And I ran into this problem yesterday already. We have several outlets, but vortexers, heat blocks, scales, all these things are just plugged in. What I really want to do is switch to power strips so we can turn everything off when it's not in use, right? Especially, right, the worst thing we could be leaving a heat block on for days at a time. The incubator, I went in and the incubator that I have is just on. I'm not using anything right now, but the incubator is just plugged in, right? Just to start thinking about these types of ideas. Same thing with the fume hoods. It's very easy if you don't close the fume hood, or if you close the fume hood to have it stay on, right? So to make sure that the fume hood isn't on when not in use. And then sterilization. We have a large commercial scale autoclave, and I need to get better about using it at full capacity as opposed to one beaker at a time. Some other things I think about definitely are cold storage. So in preparation for this webinar, I went into my freezer and tried to purge old samples to make space. And the problem, so this is a good example of the issues. You hold on to everything because sometimes you don't know when you need it. And I went into the freezer yesterday, threw out a bunch of old samples, and there just happened to be some samples that a student needed. I took them out of the trash, it was okay. But anyway, cold storage is important. Thinking about the water in the lab. And for me, field work, electronic data collection instead of paper. I thought this was a really great way of maybe saving on some resources. Finally, inventory management. So this was really important for me, setting up my new lab. I already made a mistake. If we think about ordering only the materials we expect to use. So I had this picture of pipette tips here because this is basically what I'm going to be able to do. I went way overboard on my pipette tips, and now I basically have half my lab filled with pipette tips. Fortunately, they don't go bad, but you know, if it was a product that goes bad, I didn't do a good enough job of inventorying it. So there's things to be careful of. And for me personally, gloves is a huge issue. I didn't know about these recycling glove programs, but you know, you train your students to change your gloves between protocols, to change your gloves if you need to scratch your face, to just constantly change gloves. It's such a waste, and being able to recycle them would be a really, really great step. So anyway, I just wanted to share a little bit about me, my research, working with the Westchester Biotech Project, and how my research has now informed me thinking about lab sustainability and setting up my own lab. But thank you all very much for listening.
Thanks, Stephen. That's great. We'll turn it over to Erika, and I believe Erika will talk about some practical steps that any of us can take that are in line with some of what Stephen just talked about to operate more sustainably. So, Erika?
Hi there. Can you hear me, Chris?
Yeah, that's great.
Yes, okay. I just wanted to make sure I was off mute on all platforms. So I'm actually going to start with a little bit of an introduction to NESI, the Network of Early Career Sustainable Scientists and Engineers, a little bit of an introduction to my Green Lab, which is the company that I'm currently working with, and then continue on to my presentation. We were actually supposed to have our executive director, Dr. Alex Hicken, of NESI be doing this, but unfortunately she is very ill and was not able to attend. So I'm going to try my best to do a last minute fill-in for NESI. So the Network for Early Career Sustainable Scientists and Engineers is an interdisciplinary global community of scientists and engineers who are developing solutions for a prosperous and sustainable future. So basically this means that it's a network of people from all around the globe that are trying to get in touch with each other and come together to create innovative solutions and share what they're doing in their research through this network and meet with each other and collaborate and just try to build up sustainability in science in general as this next generation of scientists move through their careers.
Hey, I just want to jump in real quick. It seems like the slides might be cut off a little bit. Stephen, what do you see? Yeah, they were kind of half. Oh, that's much better. There we go. Okay, great.
Are you seeing them now?
Yeah, now it's beautiful. Yeah,
okay.
Sorry about that. I just wanted to make sure we caught that. Cool.
Okay, no, I had a bunch of different apps open on my computer, so it must have been just the size that it was on there, but I have it on full screen. So, okay, it's good now?
Yep.
Okay, perfect. Sorry about that. So yeah, just going back to this is just the map of when I was explaining who we are and what we're doing through NESI. This is the map of where different people from the network are currently living. So you can see that we have a pretty big presence all around the world. And I should also mention that I am part of the board of directors of NESI and the director of Sustainable Science Groups. And so just to tie that back in a little bit of why I am presenting these slides. And then our approach is kind of a bottom-up interdisciplinary approach. So we're just connecting with each other around these big global challenges that we're all facing and any solutions that we've come up with to those, and trying to build up developing leaders and training leaders within this realm. and also providing inspiration, which is always great through a network to feel like you're part of a community and that you're not the only one in the world trying to do these types of things within your research. Also building communities, we host events, build partnerships, support local sustainable science groups, which is my primary role within NESI. So if anyone has a sustainable science group or would like to start one, please get in touch. And I should also mention that it's free to join, and you just go to the website and sign up today. So please join if you're interested. And now I will start with my portion of the presentation on behalf of my Green Lab on creating sustainable labs. And with my Green Lab, as Chris mentioned in the introduction, I am a program manager and work on projects all across the US and Canada. So my Green Lab was founded in 2013 in California, and we are a 501c3 nonprofit organization. We reached over 10,000 scientists in 2018 alone, and we grow every year. So it's a really exciting organization to be with, just because even though we're a small nonprofit, we have this big reach and this big impact, and people are really excited about the work that we're doing. Our mission is to build a culture of sustainability through science, and we focus on 4 different areas: so energy, water, chemicals, and end of life. We have a Green Lab certification program. So this is an online assessment program that there's 5 different levels of certification a lab can achieve. We currently have over 200 labs certified through our online assessment tool. And we have also co-founded something called the Center for Energy Efficient Laboratories. And this is really to provide financial incentives for people to purchase more energy efficient equipment. For example, one of the projects that you might be familiar with is the -80 or ultra low temperature freezer Energy Star rating that came out in the past year. We worked on that through the Center for Energy Efficient Laboratories. And if you're not familiar with that, next time you're purchasing a -80 freezer, you should definitely check the Energy Star ratings and ones that are approved. We also have an ACT label, which is an Environmental Impact Factor Eco label. And this is to standardize green scientific product labeling and facilitate procurement decisions. So if someone in a lab is trying to buy a piece of equipment, say a water bath, for example, and you're interested in looking at the environmental impact of each. So not only the electrical consumption, but how it's made, the user impact, how it's dealt with at the end of its life. You can look on the ACT website and see the 2 different labels before you make that purchase, which are independently audited from the companies and the manufacturers. And that can help guide your decision to see what's important to you, or if you're just interested in the overall lowest impact, then you can go with that rating as well. And if you have any questions, please email us. And then finally, our other kind of big branch that we work on is guiding green chemistry experiments in the higher education curriculum. So we worked with another nonprofit based in Boston called Beyond Benign, and they focus on green chemistry education in K through 12 and higher ed and teacher trainings and things like that. So we worked with them to facilitate an integration of green chemistry into teaching labs by making this comprehensive guide that I'll talk about in a little bit more detail later. So the impact of labs, as Stephen was saying, a lot of us live a really sustainable life and work on research that's about the anthropogenic effects on various either species or chemicals or things like that on our planet. But we never really think about how we're doing that, how that affects our planet and what we're studying. So just to throw out some quick facts, labs use about 5 times more energy per square foot than offices. In 2014, worldwide, there was 12.1 billion pounds of plastic waste generated from laboratories. And finally, each fume hood consumes the energy equivalent of burning over 1,700 gallons of gasoline every year. And I did a quick calculation. That's over 140 tanks of gas in my car. So that's well over a year's worth of gas. So this is consuming, you know, quite a substantial amount of energy, probably 3 years worth of gas in my car for just 1 year of using each fume hood. And, you know, multiple labs have multiple fume hoods. So, and that's why really my Green Lab is here to help. We're here to help you make a difference in the work that you're doing and the great science that all these scientists around our world are doing. As scientists, we advance medicine, design consumer products and electronics, ensure healthy crops, design fuel to feed 7 billion people. And we really need to think about doing this without causing harm to human health and the environment that we're trying to protect. And so we can all contribute to sustainable practices, not only at home, but also through Green Lab programs and initiatives by extending the environmental impact considerations that we have into the research that we're doing. So as I mentioned earlier, the labs use about 5 times more energy per square foot than offices. So there are a lot of opportunities for energy conservation in labs. The reason that labs use this much is there's a lot of energy intensive equipment. It's around the clock operations. Labs are not a 9 to 5 operating space. There's 100% outside air requirements, high airflow rates, up to 20 air changes per hour. So full volume of air in the building has to be exchanged with outside air, and that requires a lot of energy to pump all of that, purify that air, decontaminate or dehumidify it, sorry. And then also there's heat generated by lab equipment that can lead to overcompensation by the heating, ventilation, and air conditioning system, the HVAC system that I was mentioning in my last point there. And so all of these and more contribute, and also how we interact with all of these systems contribute to this high energy use. So some of the things that we can do are with our fume hoods, as Stephen mentioned, shut the sash. I mean, safety is the #1 priority, and they should be closed at all times that you are not in there doing an experiment for safety reasons. But kind of as an added bonus, when fume hoods are left open, they're using about 3.5 homes worth of energy. You can reduce that by 40% by just closing the sash on variable flow fume hoods. Biosafety cabinet, same thing. When you shut the sash, in most cases, the blower goes off, and the UV light should be turned off as well. And if the blower does not go off when you shut the sash, there's a turn off button manually. So when you're not using those, same thing with fume hoods, you should make sure that they're completely turned off and closed. ULT freezers, -80s. So there's a couple of things we can do around those. One is to chill up to -70. So instead of setting it at -80, most samples are perfectly safe at -70. And this reduces the energy consumption of that freezer by over 30% in most cases, and extends the life of the compressors, and is just providing a healthier environment for the freezer to operate, as it's not going to be producing as much heat, and just reducing energy overall. Maintaining an inventory and cleaning out old samples so that we're not purchasing unnecessary freezers to store samples that we don't actually need. And maintaining an inventory can really help with that. Things like vacuuming the coils, taking off the ice, ensuring the seals are well maintained. All of these can help not only in best practices for maintaining your freezer and saving you money, but also for saving energy as well. And then my Green Lab, in collaboration with I2SL, the International Institute for Sustainable Laboratories, has an annual International Lab Freezer Challenge where we have some pretty good prizes. So if you're doing any or all of these best practices, you can earn some credit and be recognized through that challenge. And then finally, turning off equipment when it's not in use. I know it sounds so simple, but it's often not done. Things are left plugged in and turned on just in case they're needed. And there's a couple of solutions. Sometimes they're not turned off because people don't know how to turn off that equipment properly. So going through and writing up a startup and shutdown procedure and having training on that is really helpful. And then also things like outlet timers to put that equipment on schedule. So back to the water bath example, if you have a water bath that you leave on all night because you need it hot and ready when you get there in the morning, something like an outlet timer would be really helpful to have that turn on maybe a half an hour before you get there, so it's hot and ready when you get there, but it hasn't been running all night. Also reduces the fire hazard opportunities in the lab. Also, water conservation. We're based in California, so we have drought issues, and water conservation is a really big focus in this state especially. And so there's a couple of things we can particularly address in labs. One is water miser installations on autoclaves. So not only can you run a full load, which is really important, but also there's a device that you can save over 1,000 gallons per day per unit. So it reduces the flow rate by 75 to 90% from the normal flow rate, and your autoclave still works completely fine. It just reduces mostly the things that happen in the back end, not actually the sterilization that's going on, but the cooling and rinsing and things that are happening that don't necessarily need to be happening 24/7 when it's not in use. Adding aerators onto faucets. So these are the little tiny devices or piece of hardware that's on the end of a faucet. And without one of these, your faucet can use we've found about 3.6 gallons per minute without an aerator. And you can buy aerators that are anywhere from 2 gallons all the way down to 0.5 gallons per minute. And you can install these in your home too. They cost only a couple of dollars, and they have a really substantial impact on the water usage and the flow coming out of the faucet. I've done some similar projects at various institutions in California. We've seen a reduction of over 4 million gallons of water per year just by installing these little tiny devices on the ends of faucets. So they have a really big impact for such a low cost, low time installation project. And then finally, the elimination of single pass cooling. So my background's in chemistry, so any chemist out there will recognize this distillation setup. You have what we call single pass cooling. So this is when you use a condenser and you have water coming directly from a cold water faucet in one end of the condenser. It flows through the piece of glass, up, out, and down a tube directly to the drain. So this is one single pass happening. There are setups that you can have with even as simple as a 5-gallon bucket and a fish tank pump to recirculate this water to continuously cool, but not use new water each time and be wasting that down the drain. Not only is it a flood prevention strategy, because if one of these tubes comes off, you have free-flowing water coming out of a faucet, which can cause a flood in a lab pretty quickly. But there's also savings of hundreds of gallons per day per unit, depending on how many of these you have, how long they're running. So some things as simple as, like I mentioned, the 5-gallon bucket with a fish pump, all the way up to very sophisticated recirculating systems that are installed in a building. And then beyond those specific resources, there are additional green lab opportunities, things like vendor take-back programs. Ask your local representative or sales rep that you work with if they have any take-back programs, or if there's anything specific that you think there might be a good opportunity that they can bring back to their sustainability team at their company. These typically already exist for things like foam coolers, pipette tip boxes. Corning has a really great program where they take all Corning, Falcon, or Oxygen products, their products, back for free. You just go to their website, print off a shipping label, and send all the packaging back. So check that out if you're interested. Increasing the installation of dry ice containers. So sometimes we consider the transportation and we don't want the dry ice to burn off, but we don't consider what we can do. So even taking some of this foam that comes in packaging that you're receiving and building up the insulation on the outside of your dry ice container, not only will you not have to order as much and it's not burning off, but all of the resources that go into making and getting that dry ice to you are going to be reduced as well. So we need to think about this as a system, not only the dry ice that's burning off directly in front of you or any other things that we're talking about, but the entire system of how these things are getting to you and what's happening after they leave your hands. Another example is nitrogen gas consumption in fume hoods. So nitrogen lines are sometimes left on high 24/7, and as you can see here, this blows out of the end of the manifold through a bubbler, and nitrogen gas is just blowing into your fume hood and up out the stacks at the top constantly. And not only is this a waste of nitrogen gas, but once again, considering the energy, the transportation, and everything it took for that nitrogen gas to get into your line, and now it's just blowing out back into the atmosphere. Finally, one of the things that we've heard about a lot in the past couple of years is ice pack reuse or take backs. We can definitely encourage you to talk to your vendors and sales reps about potentially taking these back for any cold storage samples that are arriving. Also, if you can't make arrangements within your institution or with your sales reps, try to look outside and set up a program. Some animal shelters, vet hospitals, we found might need these. There's a bit of an issue. I wouldn't recommend necessarily going to things like a food bank to try to take these, as they have been in a lab, but there are some places in need that will take reusable ice packs as well. Finally, I'm going to try to speed up a little bit. Opportunities for green chemistry. So green chemistry, for those that don't know, is not a subdiscipline of chemistry. It's an entire kind of overarching umbrella theme that applies to all areas of not only chemistry, but economics, engineering, philosophy, toxicology. It's governed by 12 principles. And for example, one of them is use safer solvents and auxiliaries. So thinking about the solvents that you're using, what they're doing to the environment or their impact on your health and safety, and if there's alternatives that can be used instead. Not only is there cost savings for these, there's economic benefits, and as I mentioned, safety is always a priority. And once again, industry is in need of these skills from new graduates. The GC3 has statements from companies saying that they will preferentially hire people with green chemistry skills. So this is important in general for safety and cost savings, but it's also a skill that companies are demanding from new graduates. Back to our guide. So we did create a guide for green chemistry experiments for organic chemistry labs. To begin with, we're going to be releasing more of these guides in the future years on more expansive topics in chemistry. Check it out for sure. It's a partnership with Beyond Benign, supported by Millipore Sigma. There's a lot of general resources in the beginning for teaching and research labs. If you're in a research lab, I would definitely recommend just checking out the introduction section. It's completely free to download and use. You can go to our website, mygreenlab.org, under green chemistry, and you'll find it in PDF and Microsoft Word formats there. There's, as I mentioned, the contents of the guide. There's an introduction in green chemistry and a bunch of resources in the introduction. And then we do have 10 experiment packages that have everything from the experiments, the traditional demonstration, EH&S metrics for each demonstration, a comparative analysis, TA guide, sample quiz questions. So it's a really, really comprehensive guide. And I'm almost at the end here, the 2 slides left. So I want to leave you guys with the top 3 things that you can do today. And this was a really hard list to come up with because every lab is different. I know from my chemistry lab to a biology lab to a physics lab, they all look very, very different. And so trying to find 3 things that overlap with every lab is actually not easy, but I've tried to do that. So in general, things that you can all do are, like I mentioned earlier, turn off equipment when it's not in use, especially the big things like vacuum pumps, biosafety cabinets, and then your computers can be set to go to sleep instead of a screensaver, the lights. Be Good in the Hood is our campaign for shutting the stash. So you can reduce, like I said, over 30% of the energy consumption by just closing the stash. And then creating or updating a searchable electronic inventory. So this is good in itself, but extends to, like Stephen mentioned, to not over ordering and all of the greenhouse gas emissions that come with ordering, the packaging that needs to be dealt with afterwards, saving time and money in the lab and making it easier for you to find things when you need them. Some recommended outreach initiatives, if you're not necessarily in a lab but want to help with things like that, you can always run a Did You Know campaign. What are the proper recycling procedures for things like cardboard, paper towels, styrofoam in your institution? Talk about vendor take-back programs. Address SinglePass cooling, greener solvent choices, mercury-free microscopy. And then there's also a greenhouse gas emissions equivalent calculator from the EPA that's really helpful to run a campaign on things like fume hood and biosafety cabinet usage, turning off equipment, and temperature tuning of ultra-low temperature freezers. So these are just some final suggestions for things that you can do around outreach if you're not sure, like social media, posters, brochures, speaking at events, emails, and leveraging other student and staff events as well to share the information. I will end with that. Thank you so much for your time and for tuning into our webinar. If you need to contact me, this is my information and my Green Labs information. And yeah, I'm going to hand it back to Chris and Joanne, and I want to thank them as well for hosting this webinar and Westchester Biotech Project for asking us to put this on. So thank you.
Wow, thank you. This is Joanne.
Thanks, Erika.
Amazing. I'd just like to ask one question, and then I'm going to, you may have other questions, Chris, and everybody who has a question, please enter it in the chat box. We have some time for some Q&A. Well, first of all, I love both parts of this program, and I really appreciate all the checklists and all the work that My Green Lab has done to really, clarify some of the really straightforward things that can be done. And I just would you talk a little more, and maybe Stephen, you can talk about how you think about this, and hear from both of you about the whole idea of making the case. Right, there's a certain amount you can decide yourself what to do, but in some of these situations, getting buy-in from your organization, your colleagues, your suppliers, and so on. And it's probably a little different in each case, but let's start with talking about getting support from your institution to implement some of these ideas. Maybe Erika, you want to begin with any challenges or successes you can share about that process?
Yeah, I think it's different in every case for sure. There's some institutions that are really excited and just want to do everything they can, and then there's some, I would say more specifically, people that are a little bit more difficult to sell. And it's not only on sustainability; it can be on anything and that you're trying to do any
in the
project.
Lab? I've never heard that before.
So I think that it's just a matter of sitting down and talking to these people, hearing their concerns, and then making sure that you have the data to support your claims and back those up. And we have a lot of data, and there's a lot of data out there, for example, on the Labs 21 wiki page, through our website and our certification, that we have to show that there's only not a cost savings benefit, but people, for example, in California, need to reduce their water consumption, their energy consumption, and these things are often mandated by company policies, university policies, or state policies. And there's ways to do that, and these are increasing safety. If you're using safer solvents or you're reducing fire hazards, it's not really difficult to sell someone on that. So I think it's just a matter of sitting down and hearing these people's concerns and genuinely appreciating what their problems or what their issues or concerns are, and then making sure that you can help them understand, how to move forward or how to address those concerns with the data that you have available.
Thank you. So Stephen, I know it's early days in your own lab, but do you see an opportunity to try to implement some interesting practices on a larger scale at Purchase?
On a larger scale, so right, within my own lab, it's relatively easy to try to make changes with regards to recycling or freezer temperature and electricity use, things like that. Now, moving up through the academic bureaucracy can be a little more difficult. But I think it's important, you know, for administrators, it's all about cost savings. And I think, you know, showing some of the actual cost savings that can be had by making some of these changes is the real selling point if I wanted to expand it from beyond my own lab. So when I start talking to deans and chairs and directors, that's what they I mean, they also care about the environment, I'm sure, but you know,
X dollars equals X postdocs, right?
Yeah.
Yeah, that's fantastic. Chris, did you have any questions?
Yeah, this is great. Thanks, Erika and Stephen. And you know what really gets me excited, and you guys are probably aware of this too, is this whole topic just seems like a matter of time type of topic to me. Because when I break it down, it's a win-win-win. It's a win for the environment, it's a win for individual labs and their money and their resources, and it's also a win for the institutions that are ultimately supporting these labs. And so it's really kind of a thing that sells itself, and in my mind, kind of like a why wouldn't you type of question. I guess one of the things I'm wondering, and maybe you touched on this a couple times before, is how many people are already kind of aware of some of these practices when you talk to them for the first time? Because I know that when I did research, I never thought about one of these things ever. I was just thinking about experiments, and I didn't really have the awareness wasn't there. So do you see prevalent awareness or increasing awareness?
I think as we grow and as people become more involved in, especially institutions become more involved in these programs, we do see increasing awareness. But a lot of times people just don't know how to think about it in their labs. They want to make a difference, but they just are not sure exactly how they can do that or what to do. So we'll have these conversations and they'll say, yeah, a lot of this makes sense. These are great ideas. But they just, it's like you said, it's just something easy, but that hasn't been brought up before, hasn't been brought to your attention, or someone just didn't think of it. And to be honest, when I was doing research, I also didn't think about a lot of these things. I wanted to make a difference, but it's hard to sit down and sometimes think about this. And so we're trying to share as much as we can so people don't have to reinvent the wheel in that sense. But yeah, I do think that there is more awareness in general, but I also think that there is a lot of opportunity, and there's a lot of people that don't mean to be doing that, but just aren't aware or don't know that these resources exist.
Right. And I've one a little fun thought exercise that maybe. So I'm curious, has anybody ever given you a reason why they wouldn't want to do this? Does anybody have any good reason that these things aren't the best way to operate?
I think for not overall in general, I haven't ever heard like all of this is a terrible idea. But in regards to certain points, I think there are certain things that people do have very good reasons that we appreciate. That, you know, if it's going to affect the integrity of their research or something along those lines, we want to do all of this stuff while enhancing the quality of people's research. We don't want to ever be doing something and saying, oh, well, now you can't do this, this, and this. We want to focus on all the things that they can do and that are going to make things safer and more efficient and better for them. So none of this are things that we're ever trying to ask people that we're saying, you know, you have to sacrifice this part of your research. You can't do this anymore. So it's never a compromise to research, but if there are things like if they really can't shut something off, like a mass spec, I know those are really difficult things. So we can say, yeah, we understand that's a very reasonable thing, because on the back end, it's going to take a lot more resources to start that back up and a lot more time than it would be to what would be saved from turning it off. So in certain specific points, there are things, but not overall, I would say.
Yeah, it was more rhetorical in my mind that there's nothing they could say. The research is a good point. It's a good point, but this is a beautiful vision and a beautiful mission, and I believe it's just a matter of time. So good job, and keep doing what you guys are doing.
Well, thank you. Yeah, I just want to mention one project that is, I'm hoping, going to come together in 2019. Here we are in the third quarter of 2018, but it's to do a program that focuses on sustainability and process engineering. I'm sure you're aware that in order to get something, a new therapy approved by the FDA, you have to be able to make it. And so you need to go from tiny quantities to larger quantities to clinical quantities to et cetera, et cetera. And on each one of those scale-up points, there's a whole new process developed. And once something is approved, it is very unlikely that any step of the process will be changed one bit, because that may trigger a whole lot of other dominoes. So what we're going to be doing is bringing together people from big pharma, some of the sort of regional organizations focused on both sustainability and manufacturing, and so on. So I'm just going to put a pin in that, and of course, Stephen and Erika will be talking to both of you about this, and if anybody who's listening is interested in getting involved, we invite you to do that, and the Westchester Biotech Project.org website will take you where you need to go. I just really want to thank you both. This has been very informative and fun. Would you like to say any final words? Stephen, you want to go first? Sorry.
Yeah. No, I just want to again thank you for inviting me. And I have to say personally, like Chris was talking about earlier with his research, it takes some, this is not something I regularly think about or thought about in my lab. And I was really, really happy to get into this and think about changes to make for my lab. I think it's important to spread it to other scientists and other research labs.
Beautiful. And Erika?
Yeah, I also wanted to just say thank you so much for inviting me and hosting this webinar and allowing us to talk about sustainability in labs. Similarly, if anyone wants to get involved or have your lab certified, please check out our website or get in touch with me. And also, I'll just put in a little plug. My Green Lab is having our first annual Sustainability Summit hosted in Raleigh in October. You can check out our website or email me for more details. And then while it's physically in Raleigh, we are doing an online broadcasting of it as well. So we do encourage people to, view by webinar instead of traveling just for that. So if you're interested, please check out our website. We're really excited.
Beautiful, and we'll definitely have that in our newsletter for a couple of weeks for sure. Chris, would you like to do? No, thank you. Would you like to do the sign off, Chris? Great job today.
Sure, thanks, Joanne. Thanks everybody for joining us. We are going to have this video up on our YouTube shortly over the next couple days. Again, we are an open organization. We're one of those groups that we actually want to talk to you. We actually want to meet you. So if you see anything that interests you, excites you, reach out and we'll get you involved. So again, thanks everybody for being here. Have a great day.